Well, I figured I'd start this off because this is a conversation that's been about 17 years in the process of getting to. I have a letter here that I just showed Rupert that he wrote to me back in May 1989. Back then I didn't have to use this, so it's a little different today. And I had sent Dr. Sheldrake a copy of a thesis on liquid crystal consciousness. And I figured I have to send it to somebody who was thinking bigger than the people I was working with at Stanford at the time. They didn't even want to talk about this stuff. And so I was really absolutely delighted, surprised that actually he wrote back that he even read it. So I got real excited and saved this letter for 20 years now. And he writes, Dear Bruce Lipton, Dickie passed on to me a copy of your essay on liquid crystal consciousness, which I've read with much interest. That was already a good start. And then he says, I like your evocation of the dynamic resonant qualities of the cell membranes. You make a good case for this integrative role, and this is convincing. The rest of it, well, some of my things weren't that clear, apparently, so I stopped reading at that part because that was the good part. But now we have a chance to talk about it. It's only been 17 years, so I appreciate this wonderful opportunity, Rupert.
Well, so do I. And although we've met a couple of times before, we've never really had a chance to talk. So this is really our first opportunity, and I'm really pleased it's happened. And in case you're wondering whether we've spent the afternoon going through the real conversation before we meet here, the answer is no. I flew in just a little while ago on a float plane from Canada. And so actually this is the conversation. So I was, when I got your letter and when I read your stuff, Bruce, I was very excited, in fact, because there are not many biologists who think outside the box. And it was a small number then, and I would say it's an even smaller number now.
Absolutely, but the company is wonderful, so I appreciate that very much.
Yes. Well, the way I saw it really was that having read some of your more recent things and looking at and seen one or two of your presentations, both of us started from biochemistry and molecular biology. When I was working in developmental biology, I got very interested in the idea of organizing fields, morphogenetic fields, which organize living systems, as it were, from the top down. You've made a really detailed and thorough study of the molecular mechanisms and the nature of membranes, quantum tunneling processes, and so on. So your approach seems to me more from the bottom up, at least when you're dealing with cells. Of course, once you're dealing with belief, you're dealing with very high level.
Past the membrane quite a bit.
Yes,
point, yeah.
It seems to have leaped far beyond the membrane, yes. But your stuff on membranes I find extremely congenial because I was always convinced. I had a rather eccentric professor at Cambridge who, in the tutorial system there, which when I was at Clare College, I had one-on-one tutorials with this guy. Brilliant scientist, Professor Neville Wilmer, he was called. His particular obsession was membranes. And so week after week, I got these theories of membranes from Professor Wilmer. I later discovered that nobody else really appreciated quite what he was saying.
But it was interesting, just so that people will know this, because I was at the time working on it, that most people in my field were looking at membranes like saran wrap that held the cytoplasm together and basically didn't do very much more than that. So this was a completely off base, in a sense, of conventional thinking.
Yes. Well, I was sort of raised by an unconventional thinker without knowing it. And so his thing was, he worked with a small unicellular organism called Naegleria. And Naegleria lives on bacteria, and it goes around like an amoeba, engulfing things. But if you change the ionic balance, raise the proportion of potassium ions in its environment, it completely changes. Its shape changes, it grows a flagellum, and it becomes a flagellated organism and swims around with a flagellum. And then if you change it back, it changes back again. And he showed me very convincingly, this is the same genes, it's got the same nucleus, it's got the same genetic instructions, and yet its membrane can be organized in two totally different ways. He then went on to show that most cells in our own bodies fall into these two main categories. One lot are more like amoebas, the other lot are more like ciliated cells with flagella. The two most extreme examples being egg and sperm cells. But even in our mucous membranes, we've got the ones that secrete the mucus and the ciliated cells that carry it along. So different cell types with the same DNA have totally different patterns of membrane organization. It's the membrane organization that's the key. So I would lap this stuff up. I was completely convinced by Wilmer. And then I discovered no one had heard of his theories, or not many people. And so when I found your paper, I thought, at last, here's somebody else who's got this rather holistic view of membranes. It's not all just DNA and proteins.
Well, for me, it was a real exciting part of my life because, as Rupert talked about, I came from the conventional world. I was teaching in a medical school, conventional story about how DNA controls life, the central dogma, the belief of our being genetic automatons. And what got me really into this was that I was working on cloning stem cells. And it's interesting because today we talk about stem cells, some hot new thing that just came out. And my first stem cell cultures were in 1967. And I was cloning these stem cells. And what that meant was I'd start with a single cell and then let it isolate it and then let it divide. And it would create a colony of several thousand cells, all genetically identical. And so what I found was something very, very similar, is that if I took these cells and put in Petri dish A with conditions A, they form muscle. But if I took the same genetic cells and put them in another Petri dish with a different environment, they started to form bone, or even a third dish with a different environment, they started to form fat cells. And here I am in the middle of this whole world based on genetics as a controlling factor of life, and I realized, well, these are all genetically identical. The only thing that changed was the environment, and the environment controlled the cells, and that led me on a track to identify how could the environment influence what we thought was genetic control. And that led me to the understanding of the membrane as this interface. And it was fun because what I was really finding is that the membrane was an intelligent mechanism. It would read what was going on in the world around it and then send information into the cell to adjust the biology to meet the demands of the world. And so why that became important is I started recognizing, well, the information that controls the cells is not in the cell. The information that controls the cells is from the outside. And then morphogenic fields came into my reading, and this was classic because of the pioneering effort to, he got out of the box big time when he wrote this book, because I realized most of my colleagues weren't going to even talk about it either, but I found there was a kindred spirit out there seeing answers beyond what we were looking for. And I got excited. That's why I sent my work to Rupert, because I provided a mechanism by which the fields that Rupert was suggesting existed, I was showing a mechanism to convert those fields into the molecules that make biology. So it was like, I didn't have to find the fields. He brought me the fields, and I came with a membrane. It was like perfect marriage at that point.
Yes. Well, I too. It's not yet consummated, but so I was actually I didn't realize that you'd started on stem cells, because when I was doing my PhD at Cambridge, I was working on tissue cultures, plant tissue cultures, and I had very much the same experience as you. The same cells, the same genetics, change the hormonal balance and the conditions in which they're growing. And so one lot would form callus cells just growing like wound tissue. Another lot would start forming little xylem cells, the wood cells, the ones that conduct water in plants. And you could change the destiny of these cells just by changing the chemical conditions. Somehow the membranes and the cells were responding. And in the plant, when they do this in a regular pattern that I came to the conclusion must depend on fields. How I came to that conclusion was that I made a big discovery when I was a research student. I discovered that the main plant auxin, the main plant hormone called auxin, indole-acetic acid, is actually made by dying cells. As they die, the proteins break down. As the proteins break down, the tryptophan gets broken down, one of the amino acids. As that's broken down, it gives indole-acetic acid. Any dying cells produce auxin. Your dying cells, when my dying cells, any dying cell, I tried dyeing rat liver, dyeing yeast, dyeing bacteria, dyeing plant cells. They all produce this plant hormone. And plants are full of dying cells. Every wood cell in a plant is dead. It thickens up its cell wall, and then the cell commits suicide. It dissolves itself to produce little tubes that conduct water throughout plants. That's what wood is. It's made of these little tubes. And these cells that were dying, making the wood cells, seemed to be where the main hormone auxin was being produced. And this solved all sorts of problems in botany. It was, for me, a tremendously exciting discovery. At that time, I shared the beliefs of many of my colleagues that just understanding chemicals and where they're produced and where they move would help us understand development, because they'd switch on and off genes. I then realized, after a bit of reflection, this explained too much. All plants produce auxin. All plant tissues have these dying cells. And yet, if you take, say, a hollyhock plant, the leaves and the flowers, the petals, have completely different structures, and yet they have the same veins and the same chemicals. So the chemicals alone couldn't explain it. All plants have the same chemicals, yet they all have different shapes, and what's more, different parts of the plant have different shapes. That's what got me interested in morphogenetic fields, a concept I didn't invent myself. It was already there in the developmental biology literature since the 1920s. And interestingly, the very first description of it by Alexander Gervich in the 1920s was in relation to mushrooms. And Gervich was fascinated by the way that in mushrooms, fungal hyphae grow as single threads through the soil or through rotting wood. And then these separate threads, all growing independently, when the right moment comes, grow together, and then they form the fruiting bodies like a mushroom. How on earth did these separate threads know what to do? They're all the same to start with, but some form the cap, some form the gills, some form the stem, some form the membrane at the top, the skin. How on earth did these cells know what to do so it was harmoniously coordinated with the rest? And he came up with the idea of morphogenetic fields, fields concerned with the shaping of form, morphe, form, genesis, coming into being, fields to do with the coming into being of form. I thought a very brilliant idea, and of course I'll say more about it tomorrow, but this really led me into this whole field approach to biology. Not just him, but a whole series of biologists after him had developed this idea. So did you, in your, what stage did you come up with field ideas?
Well, when I started to, it was interesting, I was still stuck in the chemical world. And I identified that on the membrane, there are these structures called receptors. And there's an interesting parallel here, which we'll talk about again maybe tomorrow, and that is that we are made in the image of a cell, actually. So that if I talk about a cell or I talk about a human, we're still talking about the same thing. So the skin of the cell is very much similar to yours in a sense. It's a boundary that contains the inside, but it also has the ability to read the environment. So as we have eyes and ears and nose and taste and all these other receptors, cells have on them the same things but in micro form in a sense. So they're reading the environment. And the truth is, from my second grade image, when I first saw cells, I saw them as sentient beings. I didn't see them as just moving around in the water. They were like the amoeba would go look at something and then back away and then move somewhere else, or the paramecium. So I saw them as people, and it turns out to be, here's a very interesting relation, if you understand, that we'll talk about at some point in regard to fractals, that we are made in the image of the cell. Every function that is in our human body is already present in every cell. Anything you can identify in here is in a cell. It's got digestive, respiratory, excretory, nervous, reproductive system. Every cell even got an immune system. And so the relevance that was really fun for me is that by understanding the nature of what the cells were reading in their environment, it changed their lives. And then I started to recognize this because I'm cloning these cells in a petri dish, and the simple thing that you've learned right away in culturing cells is sometimes the environment isn't that good when you culture them, and you put the cells into these cultures, and next thing you know they're sick and dying and they don't look very good. But I found if you take those cultures and then put them into a better environment, the cells immediately recover, grow, and start to flourish. And then all of a sudden it hit me. I said, oh my goodness, realize this, that well, we see ourselves as single individual entities. That's a misperception, because the living thing, revocation of the dynamic resonant qualities of the cell membranes. You make a good case for this integrative role, and this is convincing. The rest of it, well, some of my things weren't that clear apparently, so I stopped reading at that part because that was the good part. But now we have a chance to talk about it. It's only been 17 years, so 89. Back then I didn't have to use this, so it's a little different today. And I had sent Dr. Sheldrake a copy of a thesis on liquid crystal consciousness. And I figured I have to send it to somebody who was thinking bigger than the people I was working with at Stanford at the time. They didn't even want to talk about it. Well, I figured I'd start this off because this is a conversation that's been about 17 years in the process of getting to. I have a letter here that I just showed Rupert that he wrote to me back in May 1980. I appreciate this wonderful opportunity, Rupert.
Well, so do I. And although we've met a couple of times before, we've never really had a chance to talk. So this is really our first opportunity, and I'm really pleased it's happened. And in case you're wondering whether we've spent the afternoon going through the real conversation before we meet here, the answer is no. I flew in just a little while ago on a float plane from Canada.
About this stuff. And so I was really absolutely delighted, surprised that actually he wrote back that he even read it. So I got real excited and saved this letter for 20 years now. And he writes, Dear Bruce Lipton, Dickie passed on to me a copy of your essay on liquid crystal consciousness, which I've read with much interest. That was already a good start. And then he says, I like your